Hi, this is my presentation on Oncovin. So to start, the CHOP method is a four-drug cancer treatment. The four drugs that are involved are cyclophosphamide, and this is an alkylating agent that damages the DNA. Then you have hydroxydonorubicin, which is an intercalating agent that damages DNA by inserting itself between the DNA bases. Then you have oncovin, which prevents cells from duplicating by binding to tubulin, and you have prednisone, which are corticosteroids. So to look further into oncovin, the structure is shown up top there, and it's also known as vincristine. That's the more common name of it. And it's a chemotherapy medication that's used to treat various types of cancers, such as leukemia, lymphoma, and solid tumors. It belongs to the class of drugs known as vinca alkaloids, and the way it works is it disrupts the function of microtubules in cancer cells, and this prevents them from dividing and growing. And this is important because microtubules are essential components of the cell cytoskeleton, and so they play a key role in cell division and the transport process. And normal cells divide at a slower rate or have mechanisms to compensate for microtubule disruption. So that's why they're less affected by oncovin than the cancer cells are. So tubulin, that picture on the right just shows, it diagrams the structure of a microtubule and how the tubulin is involved in that. It is a protein that polymerizes to form the structural components of microtubules, which are essential for the proper functioning of cells. They maintain the overall structure and shape of the cell by providing structural support. They organize cellular organelles and facilitate intracellular transport. And these microtubules also generate movement because they enable cilia and flagella to beat and rotate, which allows the cells to move across surfaces. So disrupting the tubulin function by drugs like oncovin can selectively target rapidly dividing cancer cells by interfering with their ability to divide and proliferate. Tubulin in cell division is tubulin assembles with microtubules that form the mitotic spindle. And the mitotic spindle segregates chromosomes into two daughter cells, and the microtubules attach to chromosomes to align and separate the chromosomes into two sets to be distributed to each daughter cell. And motor proteins use ATP hydrolysis to move along the microtubules to move chromosomes to the opposite poles of the dividing cell. So that is how it's involved in cell division. Now, the way that oncovin binds to tubulin, it binds to the tubulin dimers, which were shown in that previous picture. And these are the building blocks of microtubules. And so it binds to a site known as the vinca binding domain, which interferes with the normal function of tubulin. And this interference disrupts the assembly and function of the mitotic spindle, ultimately leading to errors in chromosome segregation. So cells that are treated with oncovin often undergo arrest at specific points in the cell cycle, which prevents the cells from progressing through mitosis and ultimately leads to cell death. So in conclusion, oncovin binds to tubulin and inhibits the microtubule formation, which is crucial for the cell division. And by preventing proper spindle formation, it disrupts chromosome segregation during mitosis. And again, cancer cells are more susceptible compared to normal cells because of how they're rapidly dividing. And oncovin is used in chemotherapy, and its interference with tubulin function contributes to its effectiveness against various cancers, making it a very common treatment method. And these are my sources.